Photosynthesis is the synthesis of complex organic materials, especially carbohydrates, from carbon dioxide, water, and inorganic salts, using sunlight as the source of energy and with the aid of chlorophyll and associated pigments. The chemical equation for photosynthesis is 6CO2, also known as carbon dioxide, and H2O, also known as water. With the help of light energy, it yields C6H12O6, also known as organic molecule, and 6O2, also known as oxygen. The process of photosynthesis takes place in the chloroplast. A chloroplast has a double membrane, an inner and an outer. Inside the chloroplast, there are thylakoids. Stacks of thylakoids are called granum. The stroma surrounds the thylakoid. The thylakoid consists of a double membrane. This is where the first step of photosynthesis takes place. That step is called light reactions. In the thylakoid membrane, there is a photosystem 1 and a photosystem 2. A photosystem is a unit of several hundred chlorophyll molecules and carotenoid pigments molecules in the thylakoid membrane. Chlorophyll A and B are the most common types of chlorophyll. Chlorophyll A is directly involved in the light reactions of photosynthesis and is in the reactant center. Chlorophyll B assists of chlorophyll A in capturing light energy, and therefore chlorophyll B is called accessory pigment. Carotenoids is a light-absorbing compound that functions as an accessory pigment in photosynthesis. It consists of yellow, orange, and brown color. The electron transport chain is a group of compounds that pass electrons from one to another via redox reactions, couples with the transfer of protons across a membrane to create a proton gradient that drives ATP synthase. The role of NADP plus reductase is to transfer the two electrons from photosystem 2 to NADP plus, so it can be reduced as NADPH, gaining a hydrogen atom and going to the Calvin cycle as a reducing agent. The ATP synthase makes ATP by adding a P to ADP. This reaction is driven by the movement of the electrons from inside the thylakoid to the stroma.
Light-dependent reactions, or light reactions, are the first stage of photosynthesis, the process by which plants capture and store energy from sunlight. In this process, light reactions is converted into chemical energy in the form of the energy-carrying molecules ATP and NADPH. In the light-independent reactions, the formed NADPH and ATP drive the reduction of CO2 to more useful organic compounds such as glucose. However, although light-independent are, by convention, also called dark reactions, they are not independent of the need for light, for they are driven by ATP and NADPH products of light.
Visible light is absorbed by pigments, which are found on photosystems. Some absorb certain colors more strongly than others, hence affecting the rate of photosynthesis. There are many types of pigments, but chlorophyll A and chlorophyll B are the most common. Chlorophyll A absorbs less blue light, but more red light than chlorophyll B, and is directly involved in light reactions. Chlorophyll B is absorbed more blue light and less red light. Chlorophyll B assists in the capturing of light energy and transferring the energy to chlorophyll A, and is thus called an accessory pigment.
The three substances that are produced from the dissociation of water molecules in the light reactions are oxygen, hydrogen, and electrons. Oxygen goes to the stomata and it produces O2 in our air. Hydrogen goes to the lumen and produces ATP used in the Calvin cycle. And electrons goes to the chlorophyll A and the ETC and it produces NADPH. The splitting of water inside the thylakoid releases electrons, which replace the electrons that leave photosystem 2 when it is illuminated.
NADH is an energy-storing compound that is found throughout the process of photosynthesis. NADP plus gathers electrons in hydrogen atoms as they come out at the end of the ETC. They then form NADPH. In the Calvin cycle, NADPH gives hydrogen to biphosphate PGA, forming G3P. The newly formed NADP plus returns to the Calvin cycle.
Chemiosmosis is the movement of ions across a selectively permeable membrane down their concentration gradient. This results in the production of ATP. The Calvin cycle takes place in the stroma of the thylakoid and involves 3 major steps. Step 1. CO2 combines with RUBP to form 2 molecules of PGA. Step 2. Each molecule of PGA is converted into a molecule of PGAL. Step 3. Most of the PGAL is converted back into RUBP, but some PGAL can be used to make a variety of organic compounds. Rubisco is the enzyme that attaches each carbon dioxide molecule to a molecule of RUBP, beginning the Calvin cycle.
3 CO2 molecules enter the enzyme Rubisco. Soon, 3 RUBPs enter as well. With the help of Rubisco, CO2 and RUBP are combined to form 3 6-carbon compounds, which immediately split into 6 PGA.
Once the 6 biphosphate PGAs have gained a hydrogen from the 6 NADPHs, they go on to form G3Ps, or PGALs. Their equation is C3H6O3. One PGAL goes out to make half a carb, while the 5 other ones go on to form RUBP. The 2 products that the light reaction creates are ATP and NADPH, which go to the Calvin cycle. ATP is made through chemiosmosis, the production of ATP using an ATP synthase to channel hydrogen from the lumen down its concentration gradient. This creates ADP, plus a phosphate, which equals ATP. NADPH is made through the use of an NADP plus reductase. NADP plus grabs electrons at the end of the ETC and hydrogen atoms to form NADPH. NADPH then goes on to the Calvin cycle.
Proteins, fats, and carbs are different types of macromolecules that can be created in the Calvin cycle. The rate at which a plant can carry out photosynthesis is affected by the plant's environment. 3 of the most important environmental influences are light intensity, CO2, and temperature. The rate of photosynthesis related to CO2 and light intensity in water is when carbon, light, and water increase up to a certain point. The rate of photosynthesis will increase. However, after the saturation point, it doesn't matter how much more carbon, light, or water, the rate of photosynthesis will stay the same. The rate of photosynthesis related to temperature is when the temperature increases, the rate of photosynthesis will increase to a certain point. After that point, proteins will denature, causing them to break down and shut down.